Hello, students. We're going to now finish our discussions on immune system disorders with the topic of immunodeficiencies. An immunodeficiency is any disorder in which there is a problem with the immune system, obviously, but specifically it could be either problems with something in the innate immune system or in the adaptive immune system, or both. And as you can imagine, this is going to result in major problems with all kinds of opportunistic infections that might even lead to cancers. We will focus on issues of the adaptive immune system branch, to where we will look at immunodeficiencies that stem from just problems with B cells, so the humoral branch. We'll take a brief look at problems with just T cells, and then the more severe form of immunodeficiency is when both branches of the adaptive immune system are affected. We will not discuss all the different problems that might arise in the innate immune system, such as problems with the complement and phagocytes, just to name a few. That said, we can classify immunodeficient disorders based on whether they are genetic disorders that are literally inherited, or they might be present at birth and therefore develop de novo, meaning it's a new mutation. Or immunodeficiencies can be categorized as being secondary, meaning that they're acquired in life. And that's where we will primarily focus on AIDS. Now, there are many more acquired immunodeficiencies, but that will be our focus. Regarding the primary immune deficiencies, we'll first focus on the B cell immunodeficiencies. And before we get further into it, let's discuss what happens to antibodies in the fetus and child. So the first few 6 months of pregnancy, a fetus will receive antibodies from its mother. So these IgGs are immunoglobulins proteins that are small enough to cross the placenta. The IgMs, for instance, are not. They're just too big. And then after about 6 months, we're going to see that the maternal IgGs are beginning to decrease, because this is the time that the fetus can start producing its own first immunoglobulins. And remember that it's always IgM that's first produced, and later on it can go through class switching to produce some of the other antibodies. So within a couple of years or less, a child has pretty much reached the levels of immunoglobulins that you and I as adults have. So an example of a primary B cell immunodeficiency is where we see that there's a problem with the maturation process of B cells in the fetus, to where they begin to produce antibodies too early. And often this is due to some kind of an infection in the uterus, which triggers this problem. What we then see happening is that instead of the fetus having mostly IgG immunoglobulins, we begin to see too high levels of some of the other immunoglobulins. Another good example of a primary B cell immunodeficiency is called selective IgA deficiency, and I will typically abbreviate it as follows. Now, this is also the most common primary immune deficiency. It is a B cell immunodeficiency, but even if we look at all primary immunodeficiencies, this is the most common. And it's, of course, as the name says, a deficiency in IgA, so we can refer to it as a type of hypogammaglobulinemia. Who can say that word? So too low levels of a particular immunoglobulin in the blood. Remember, emia refers to blood. So what causes this problem? It's a problem that is inherited, and how it is inherited can really vary, but specific chromosomes have actually been identified. So literally what happens is that B cells that normally produce IgA are not maturing properly, and consequently, the person ends up with either hardly any IgA in their serum or very low levels, particularly in persons older than 4 years. So this starts pretty early on in life. Now, the other immunoglobulins, particularly the ones that play a really important role in immunity, such as IgG and IgM, they are occurring at normal levels. So because kiddos who suffer from this particular immunodeficiency do tend to have enough of the other immunoglobulins, it's thought that that explains why most um, kids that have this condition are actually asymptomatic. But of course, some do have symptoms. And as you can imagine, if there is a lack or a low level in IgA, which is the immunoglobulin of your mucosae, right, we're going to see major infections of various body tracts or possibly all body tracts. In addition, people might have an increased amount of allergies, they might develop cancers, or even different types of autoimmune disorders. This can occur in both males and females. So let's take a look at an example of a T cell immunodeficiency. As you can see, we're just touching on each one of these immunodeficiency just briefly, just to get the point across of how they work and what kinds of consequences they can have. We're not really going to list many different examples of each one of these immunodeficiencies. So for T cell immunodeficiency, I've picked the George syndrome as an example. And here the problem is that the child really does not form a proper thymus. So we see something called thymic hypoplasia. Actually, that's what the synonym is for the George's syndrome. And as you can imagine, if there is no well-formed thymus, how are the T cells going to develop properly? Again, this is going to be present in both males and females, and like all of the primary immune deficiency disorders, it is a very rare kind of condition. So while the George's syndrome can be inherited, it most often is just a sudden de novo, as we will call it, a brand new mutation that occurs in a particular chromosome. This tends to happen during the time of meiosis, when either a sperm cell is produced in dad or an egg cell is produced in mom. So the inherited form, which is going to be an autosomal dominant genetic condition, is quite rare. But let's take a quick look at it and do some review of genetics. Remember, when a trait is autosomal dominant, it has a very high chance of being inherited. So here we have dad who is totally unaffected. So both of his chromosomes are carrying the gene that does not trigger the George's syndrome. Mom only has to have one gene on one of its chromosomes, I should say, while the other chromosome is healthy, for her to create problems possibly in her offspring. And so notice that, of course, there's no guarantee both these parents will produce 4 children, but we always look at chances, right? When we look at a couple having children. So they have a 50% chance that they will produce children with the disorder, and this is all because each parent must always pass on one of its chromosomes. So this child received both healthy chromosomes from each parent. This child, on the other hand, received a healthy chromosome from dad, which is the only thing he can donate, while mom ended up donating via her egg one of the unhealthy or I should say mutated chromosomes, and consequently that child will be affected. So if you go through the possible gametes that each one of the parents can produce and make them into possible zygotes, you will find that almost that 50% of their offspring could have DiGeorge's syndrome. So pretty high incidence of, or pretty high chance of inheritance there. So in these fetuses that are afflicted by DiGeorge's syndrome, we're going to see that the problems develop pretty early on, or before the 12th week of gestation, and that's the time when the thymus really starts to develop. Now, it's not just the thymus that is having issues, it's that whole region around the thymus, which includes the parathyroid glands, even parts of the head, neck, and heart, because your thymus, remember, covers a good portion of your heart. So that whole region is impacted, and that has consequences. Let's first talk about the consequences regarding the thymus. Clearly, this child is not going to produce healthy T cells and is going to consequently deal with all kinds of fungal and viral infections constantly. In addition, because the parathyroid glands are not developing properly, if at all, the person or the patient is going to have issues with PTH levels, which stands for parathyroid hormone. And parathyroid hormone regulates our calcium levels, calcium concentrations in our blood. And so therefore, if we do not have enough of that hormone, we're going to suffer from something called hypocalcemia, too low calcium in the blood. And this can then result in muscle contractions or tetany, especially right after birth. Some other really characteristic consequences of carrying DiGeorge's syndrome is the widely set eyes, even low set ears, as you can see here. And the jaw seems to be underdeveloped, might not be as prominent here, but very characteristic as well for DiGeorge's syndrome. There's a relatively easy fix for this issue. I should be cautious saying that, but this can be treated by providing the child with a whole new thymus. That brings us to our last type of primary immunodeficiencies, and that is a combined form, meaning that both the B cells and the T cells are impaired. So again, this is going to be related to the adaptive immune system, often due to one or more gene problems. So as we get towards the end of our primary immunodeficiencies, we're finally going to then focus on combined immunodeficiencies where both arms of the adaptive immune system are afflicted or impacted. And the example that we're going to use is called SCID, which stands for Severe Combined Immunodeficiency, the most severe form of primary immune deficiencies. It's nicknamed the bubble boy disease. Some of you might recall this. You'll see why. And because these are going to be patients with problems in both arms of the adaptive immune system, as you can imagine, these patients are going to be extremely vulnerable to all kinds of infections and cannot live very long or could not live very long. We have some treatments for that now. So in the late 70s, a little kid by the name of David Vedder was born with SCID and had to live in a very isolated environment, and people who then visited him had to be completely covered up because he was so prone to infections. You can see here how they had to dress him in the event others were not protected, and he was very often living in a bubble when he didn't have any protective gear on. So from there, his nickname, Bubble Boy. As you can see, he didn't live long. Considering we didn't have any treatments at that time, he still lived 13 years, which is quite impressive. So like any primary immunodeficiency, there's some problem with a gene or more than one gene. And in the case of SCID, once again, these genes, these malfunctioning genes can either be inherited or they can develop anew, de novo. There's different ways in which the condition could be inherited. We'll take a first look here at the X-linked version, which is the most common. So this is the most common way in which SCID is inherited. It leads to T cells that are non-functional. Also, the natural killer cells are not functioning. We have B cells, but they really don't function well. Remember, we have impacts, both arms of the adaptive immune system are impacted. And the genetic mutation really lies in the fact that the cytokine receptors or some of the cytokine receptors are changed because of the genetic mutation so that the intracellular communication between the immune cells is not working properly anymore. On the other hand, if the condition or if SCID is inherited as an autosomal recessive condition, this time we see that there's a whole bunch of metabolic products that cannot be removed and they accumulate, which then triggers apoptosis in the cells that are developing in the thymus, the T cells that are developing in the thymus, and even the fully developed lymphocytes, both T and B lymphocytes, even the natural killer cells. So once again, we see that the T cells are mostly the problem, and the B cells sort of okay, or in number, I should say, but they again don't function well. So you see how we can have enough numbers of either T cells or B cells, maybe even natural killer cells, but just because they are present in number doesn't mean that they're functioning properly. So we have problems with the accumulation of metabolites, which triggers apoptosis of our cells, our T lymphocytes in particular, and even our natural killer cells. But we also see that there's problems with the ability to repair DNA. So if we take a quick look at the typical clinical manifestations of SCID, we see that they are almost identical to AIDS, and our next topic is going to be AIDS. So I'm not going to go into a whole lot of detail here because we will touch on those somewhat, but notice that there are going to be problems with all kinds of infections, whether they're fungal, whether they're viral, even bacterial. And often these infections are going to end up in sepsis, which is, of course, a medical emergency. In this day and age, we can provide patients with SCID a hematopoietic stem cell transplant, and if it takes, then these patients can live a much longer time than Bubble Boy did. Notice that these symptoms begin to appear very early on, around 3 months. So I thought I'd share this chart or flow chart, I should say, with you. Not that you have to at all memorize this. And you'll see that there are additional primary immunodeficiency listed that we didn't discuss. But just to give you a bit of an idea of where some of the problems lie to then trigger the problem in immunodeficiency. So on this side, we see the B cell immunodeficiencies, while on this side, we see more of the T cell immunodeficiencies. On the T cell side, we looked at DiGeorge syndrome. On the B cell side, we looked at particularly IgA deficiency. We did talk about how fetuses might have too high levels of some of the antibodies that they really shouldn't have high levels of, because a fetus and a newborn should mostly have just IgG antibodies, which it received from its mother. And then, of course, SCID is going to be appearing on both sides, although it's not really showing, well, it shows it here on both sides, exactly, because, of course, SCID is a combined immunodeficiency, meaning both arms of the adaptive immune system are impacted.